Hello, everybody, and welcome back to a new ReMicS meeting. This is Marius, and today I'm very happy to introduce Anne-Marie Aas. She has been part of this network since we started out, and she has been interested in the gut microbiota and diabetes for a long time. And today we will hear exciting data on the effect of prebiotic fibers on gut microbiota and hormones regulating blood glucose and appetite in type 2 diabetes. I'll just inform you all that this presentation is going to be recorded. If you have any questions, you can just tell them on chat, and I'll try to pass them on to Anne-Marie. So please, Anne-Marie, the word is yours.
Thank you, Marius, and thank you for the nice introduction and for giving us this opportunity to present the results from the Fiberdia study. I am the project leader of the Fiberdia study, but we have many collaborators, and I'm presenting this on behalf of Eline Birkeland, who is currently finishing her PhD work on this project, but we're unable to present herself today. So most of the slides is prepared by her. So in the Fiberdia study, we are investigating the effects of prebiotic fibers on gut microbiota and hormones regulating blood glucose and appetite in type 2 diabetes. Studies on microbiota report a moderate degree of dysbiosis in individuals with type 2 diabetes. It has less of butyrate-producing bacteria, which are bacteria that have protective effect on the intestine and the intestinal barrier. The microbiota has an increase in pathogenic bacteria and in functions related to oxidative stress response. And a dysbiotic microbiota like this is suspected to contribute in the development of low-grade inflammation in several tissues, and possibly in the onset of type 2 diabetes. An increase in circulating levels of lipopolysaccharides has been detected as well. This is, as most of you probably know, a component from the cell wall of pathogenic bacteria and is considered to be caused by increased permeability of the intestine. Some studies show reduced response of the incretin gut hormone, GLP-1, in type 2 diabetes, and others do not. Dietary fibers that escape digestion in the small intestine are fermented into short-chain fatty acids in the colon by the microbiota. If the dietary fibers are fermented by bacteria that are considered healthy, they are classified as prebiotics. So in short, prebiotics are food that are good bacteria, the good bacteria in the colon. The short-chain fatty acids are important sources of energy for the colonocytes and are used as substrate for gluconeogenesis and lipogenesis in the liver. Over the last years, the short-chain fatty acids have also been identified as important signaling molecules. They bind to the G protein coupled receptors, GPR41 and GPR43, resulting in various effects depending on which cells are affected. In the enteroendocrine L cells, it results in the increased production and secretion of the gut hormones PYY, GLP-1 and GLP-2, which are released in response to feeding. PYY is a satiety hormone, and GLP-2 improves the barrier function in the gut. GLP-1 is an incretin hormone, and it binds to receptors in different tissues. So in the pancreas, it enhances secretion of insulin in a glucose-dependent manner. It has a protective effect on the beta cells, and it inhibits release of glucagon. GLP-1 also promotes satiety, both by direct effect on the appetite center in the hypothalamus and by delaying gastric emptying. GLP-1 is rapidly deactivated by the enzyme DPP-4, and both GLP-1 analogs and DPP-4 inhibitors are used in the treatment of type 2 diabetes. The health benefits of prebiotics have been studied in healthy individuals and in patients with various diagnoses. Amelioration of dysbiosis, increased response of GLP-1 and PYY, improved insulin sensitivity, increased subjective feeling of satiety, and reduced energy intake are some of the results from clinical trials. But despite the obvious benefits of improved regulation of blood glucose and appetite in type 2 diabetes, the effects of prebiotics have hardly been studied at all in this population. So that is what we aim to do. And our primary outcome measure is GLP-1 response to a mixed meal tolerance test. And the secondary outcome measures are glycemic regulation, regulation of appetite, microbiota composition, and fecal short-chain fatty acids in this population of type 2 diabetic patients. We chose a prebiotic supplement consisting of a 50-50 mix of oligofructose and inulin. Inulin is a multiple molecule of D-fructose chained together with one molecule of D-glucose attached at the end. Oligofructose has less than 10 molecules of D-fructose in the chain. They are also commonly called Inulin-Type fructans. We decided on Inulin-Type fructans as choice of prebiotic fibers, as these are the most studied among prebiotics, and a mixture of both long and short chain inulin have been proposed to minimize the expected gastrointestinal symptoms. The participants consumed 16 grams per day for 6 weeks. And if you want to ingest 16 grams of these prebiotics from a natural food source, you can eat 60 bananas or 2 raw onions or 320 grams of asparagus. For placebo, we chose maltodextrin, which is the most commonly used placebo in comparable trials. Here you see the crossover design illustrated. All participants have 6 weeks on prebiotics and 6 weeks on placebo, but some start with prebiotics and some start with placebo. In between the 2 periods, they have a 4-week washout without any of the treatments. Before and after the 6 weeks periods of intervention, the participants spent the day at the hospital for testing. These 4 visits, the participants arrived in fasting state early in the morning. They delivered food frequency questionnaires and samples of feces for analysis of microbiota and short-chain fatty acids. Before noon, we performed a mixed meal tolerance test. The participants then consumed a liquid meal of nutrient drinks. Blood samples were obtained in fasting state and at 8 time points after finishing the meal. And in the afternoon, we performed a test lunch. The participants were served a casserole of pasta with meatballs. It's a Fjordland-rett. They were instructed to eat as much as they pleased, and all food eaten were weighed. Subjective feelings of appetite were measured before the meal and after, and at 4 time points after the meal by using visual analog scale. Questions about their appetite were answered by drawing a vertical mark on a 100 millimeter line. So you're supposed to rate your hunger according to from this left end. You had the most extreme negative. I'm not hungry at all, and on the other side, I have never been more hungry in my life. And then you put down the point where you think it is. It's most likely to end. Um, 131 individuals were assessed for eligibility. and 36 of these were randomized to start with either prebiotics or placebo. In total, 25 participants completed the intervention. Individuals using insulin or GLP-1 analogs were not included in the trial, and the participants stopped taking diabetes medication 2 days in advance of each of the 4 test days. Of those who were randomized but did not start or complete the intervention, 3 had to take antibiotics, 3 were diagnosed with serious illness not related to the intervention, and the rest were excluded or withdrew for other reasons, but not due to side effects of the treatment. The compliance was excellent, with only 3% of the prebiotic supplement packages returned and only 4% of the placebo supplements returned. So these are the baseline characteristics. As a group, they were overweight, 40% were women, and they had a diabetes duration of approximately 5 years. Nearly 1/3 of the participants did not use any blood glucose lowering medication, while more than half of them used metformin, a drug that has been shown to affect the gut microbiota. However, in our study, the metformin dose was kept unchanged, and we found no difference in the overall fecal microbiota between participants using metformin or not. Apart from the reported higher intake of dietary fiber, the subject characteristics showed a population quite typical of patients with type 2 diabetes in Norway. So these are results from the microbiota analysis performed with 16S rRNA gene amplification. The analysis were done by Ida Ruud and her group at Nofima Research Institute. And the biostatistical analysis were performed by Ingrid Måge, also from the Nofima Research Group. The results from this part of the study was recently published in the European Journal of Nutrition. The data show abundant inter-individual variability of microbiota composition, as illustrated here with an overview of the inter-individual variation of phyla at baseline. As you can see, there's a gradient distribution of the dominating Bacteroidetes shown in orange, with a mean abundance of 69%, and Firmicutes shown in green is the second dominating phylum, with a mean abundance of 26%. Indeed, except for these two participants, Bacteroidetes accounted for more than 50% of the microbiota present in the individuals. The other phyla shown here were present to a variable degree between individuals. We found no significant effect on alpha diversity of neither after prebiotic treatment or the placebo treatment. And the prebiotic effects on microbiota composition in the present study was moderate, accounting for only a few percentage of variation in the microbiota. Only 2.2% of the total variation at phylum level and 1.5% of the at OTU level. The overall effect did not reach significance at the phylum level, although Actinobacteria was significantly positively affected by prebiotic fibers compared to placebo and baseline after 6 weeks of intervention. This figure shows the increase in relative abundance of the Actinobacteria phyla after 6 weeks of prebiotic treatment compared to placebo. The prebiotic fiber had significant effect at the OTU level, however, with significant impact on 32 OTUs. These are presented in this figure with the representative effect sizes. The OTUs affected by the prebiotic interventions are sorted by effect size. That is the difference between prebiotic intervention and prebiotic intervention period compared to placebo period and baseline. So dominating OTUs are indicated here in bold. You can see some of them. And the relative average abundance of the OTUs is included at the right in this bar here. Bars are colored according to representative phylum. So the 3 OTUs with highest positive effect sizes were of actinobacteria and assigned to Bifidobacterium adolescentis. The Bifidobacterium bacteria are not able to produce short-chain fatty acids themselves, but many of the species from this genus are capable of degrading the chains of Inulin-Type fructans, and in this way prepare for other species to complete the fermentation and produce short-chain fatty acids. This process is called cross-feeding. High levels of bifidobacteria are associated with a healthy microbiota, and bifidogenic health benefits are of particular interest in type 2 diabetes, have been shown in several studies. Trials in humans and mice report that bifidobacteria may prevent endotoxemia and improve regulation of blood glucose. Bifidobacteria trials has also shown anti-carcinogenic properties, positive effects on blood lipids, and reduced symptoms and maintained remission in inflammatory bowel disease. The remaining OTUs positively related to prebiotic fiber intake were not that highly ranked and with less effect sizes, and were mostly of Bacteroidetes origin, shown here in yellow, and firmicutes, shown in green. Especially OTUs within Bacteroides were among these, including one dominating OTU assigned to Bacteroides ovatus. I'm not terribly good at pronouncing these names, neither in English or Norwegian, but I hope you can read the screen. Yes. Yes, and 3 OTUs within Clostridales, including Lactospiraceae and Faecalibacterium prausnitzii. The latter species were also increased. And this prausnitzii is known to be able to degrade the chains of inulin-type fructans, as well as produce the short-chain fatty acid butyrate. The OTUs that decreased with the prebiotic fiber were firmicutes, including dominating OTUs assigned to the families Rubinococci and Lactospiraceae, all with high effect sizes. And in addition to this, there's one that I can't pronounce at all, this one, that declined with the prebiotic fiber. We also measure the fecal content of short-chain fatty acids as indication for microbial activity. The analysis were performed at Unge Vetlesens Institute, Lovisenberg Diakonale Sykehus, by Gunn Helene Malmström and Jennifer Finnes in Jørgen Valør's research group. Acetic and propionic acid have been linked to mechanisms preserving or improving glucose homeostasis and appear to be anti-carcinogenic. And propionic acid is able to reduce visceral and liver fat, which both are important to reduce insulin resistance in type 2 diabetes. Butyric acid is of particular interest in type 2 diabetes, as animal studies report it improves glucose homeostasis by inducing gut production of GLP-1 and PYY, as well as protecting the gut barrier function. The most abundant short-chain fatty acids, acetate, propionate, and butyrate, are commonly present in feces in the approximate molar ratio of 60-20-20. And as expected, this was also the case for the fecal samples from our participants. In all the following figures, the prebiotic period is depicted in blue and placebo period in red, with baseline in lighter colors and 6 weeks after in darker colors. For the total amount of short-chain fatty acids, there was an increase in the prebiotic period that was significantly different from the change in the placebo period, as shown here. An increase in the prebiotic period and no change in the placebo period. We also found increased levels of acetate and propionate after prebiotic treatment that was significantly different from the placebo treatment. But the increase in fecal butyrate did not reach statistical significance, even though there is an increase, as you can see here, from baseline to 6 weeks in the prebiotic treated group. The lack of significantly increasing fecal butyrate could mean that we didn't have strong enough statistical power to detect this, or that there really wasn't an increase. But it could also mean that the butyrate already had been used as an energy source by the colonocytes before it reached the distal colon, as we analyzed this in the fecal samples. The relationship between microbiota and the short-chain fatty acids is presented in this heat map, only including the OTUs significantly affected by the prebiotic intervention and sorted by their effect sizes, just as in figure 1 that I showed you earlier. And dominating OTUs are indicated in bold here as well. And correlations is estimated with Spearman's row coefficient, where red is positive and blue is negative relation. Asterisk indicates significant relationships. A general trend was that acetic acid was positively related to OTUs that increased with the prebiotic fiber, as you can see here. That means the opposite trend was observed for the OTUs that declined with the prebiotic fiber, as you can see here. These are those who declined, and there's an indirect relationship. That means as these species increased during prebiotic treatment, the acetic acid content in the fecal samples also increased. Whereas while these OTUs decreased during prebiotic treatment, acetic acid content increased. Interestingly, the prebiotic affected OTUs of Bifidobacterium adolescentis were negatively related. Let's see, there we got it here, to butyric acid. And only Lactospiraceae was positively related to butyric acid among the prebiotic affected OTUs. That's here. Now on to the main outcomes of the study, the effect of these prebiotic fibers on glucose regulation and appetite. These outcomes were measured during a mixed meal tolerance test. And the following figures have the same color codes as the previous, with the prebiotic period depicted in blue and placebo period in red, and with baseline in lighter colors and 6 weeks after in darker colors. And the prebiotic intervention had no significant positive effect on our primary endpoint, the GLP-1 response. In fact, when we analyzed in a linear mixed model, there was a 4.8% reduction in plasma GLP-1 concentrations after 6 weeks of prebiotic treatment, and an 8.6 increase after the control treatment. Still, differences between and within the two treatments in estimated areas under the curves were not significant. There was no significant effect of prebiotics or placebo on GLP-2 response. And for blood glucose, we found no significant effect either, neither of prebiotic intervention or placebo, and not for insulin either. For the hunger hormone ghrelin, we found no significant effect of the prebiotic supplement over placebo. However, the two treatments showed a significantly different impact on plasma concentration or total PYY during the standardized meal. PYY response increased significantly after control treatment, but remained unchanged during the prebiotic treatment. So this was opposite of our hypothesis. So did this small effect on the placebo treatment on the satiety hormones GLP-1 and PYY show in subjective ratings of appetite during the ad libitum test lunch? This figure shows the mean portion sizes that participants consumed at the 4 test lunches. And as you can see, there were hardly a spoonful differing between the 4 test days. They ate approximately 450 grams of the casserole dish before and after both prebiotic supplementation and placebo supplementation. And for the feeling of hunger, we found no significant effect of prebiotic over placebo. As you can see, subjective feelings of hunger decreased rapidly during the first 30 minutes and then increased gradually during the next 2.5 hours. But there is a remarkable similarity of the ratings done before and after both prebiotic and placebo treatment. And the same goes for the ratings of satiety, except that the curves goes in opposite direction. So in summary, we found some significant positive effects of the prebiotic supplement over placebo on the microbiota and fecal short-chain fatty acids. But we were not able to detect that this had a further positive impact on GLP-1 response, glycemic regulation, GLP-2 and appetite hormones, or the subjective feeling of hunger and satiety. Comparing the present trial to similar studies where significant effects were reported, our sample size was equal to or higher than most of the other studies, and the duration of the intervention period and washout period should suffice, as well as the supplement dosage. So perhaps the effect of microbiota wasn't strong enough to increase the response of gut hormone, or perhaps supplements with Inulin-Type fructans just don't have the same therapeutic potential in type 2 diabetes as reported in other populations. So with that, I leave you with a picture of our collaborators. Quite a lot of them, as you see here, and thank you for your attention.
Thanks a lot, Anne-Marie. Very nice presentation. We have been following from ReMicS Hospital, and we have a few questions. It's really so this is really a negative study when it comes to your predefined endpoints. It's interesting to see the effect on the microbiota composition and then the potential effect on the short chain fatty acids. I think it was interesting to see the heat map that you showed. That it seems that butyrate are correlated in an opposite pattern compared to acetate and propionate.
Yeah.
Could you comment on that? I mean, the overall response on short chain fatty acid is, as you say, positive, but it seems that they vary a lot. And it is from the pattern here. Acetic acid and propionic acid, they have the same colors and very often the opposite color as butyric acid.
Yes, and butyric acid was the one that really aimed to change. But
yeah.
so no, of course there is some limitations with these measurements of the short chain fatty acids that we can speculate about, because of course we're not really measuring what's in the gut, but rather what's left in the fecal samples when it has passed through the intestine. So it might be a problem with that, but still there's a tendency, and we have significant correlations and trends, but there's also very much a variability both in the prebiotic effect of the fiber, how it affects the microbiota, and also a big variability in changes in short chain fatty acids. So I don't really have a very good answer, but if you have suggestions, I would like to hear.
Did you make any attempt to predict the genetic? Sorry, you had two computers in the same room.
Yeah. The genetics.
Did you do any attempt to predict genetic potential of producing butyrate?
You think the genetic of the bacteria, or?
It could be possible to predict with pie crust bacterial genes from your bacteria or microbiota data, and then there is a limited enzyme for butyrate production, for instance.
Yes.
Use that or that and try to correlate that with the production. Just that will give you at least a strong association between microbiota composition and the butyrate production potential.
Right. No, we haven't done such analysis, but that it sounds like a good idea. These data are, as I said, they are published now, but still it could be interesting to do such analysis.
I just have the feeling that acetate and butyrate use the same rate limiting enzyme, but I think we have to look it up. Yeah, I mean in Pycrus, but yeah.
I don't know. That would just be one way of moving a bit further, just to dig in your data.
Yeah, thank you for that suggestion. It's something we should do.
Just an overall question. I think in your conclusions, you said that this intervention had a positive effect on the microbiota composition. Could you please define what is a positive effect on microbiota?
No, yeah, that's always something you could discuss also. But what we mean with that is really the increase in the bifidobacterium that has been, as I said, connected to several health benefits. So that we think we can say that there's a positive effect. And there is an increase in this Prausnitzi. I can't, I'm so terrible at pronouncing this, but that's one OTU that is been said to have, been good at producing butyrate and affect the endpoints that we were looking at.
Yeah. And still there is a lot of Lactospiraceae and Ruminococcaceae that are decreased, that are also supposed to be butyrate producers, but I guess it's only supposed to be. I guess the data show that maybe they do not produce butyrate. But have you considered doing a full metagenomic analysis on this to go down to species level in your data set?
Well, No, we haven't done. We had well, we don't have the economy to do that, I think. But we do have samples, so it is possible to do it, I guess.
Yeah.
But it's an economical question, I think.
Yeah, sure. There's a question here from Jonas André Lundekvam, where the fecal samples for short-chain fatty acids analyzed fresh frozen.
Yeah.
Yeah, thanks.
They were fresh frozen, yes.
If there are other questions, I think you can just ask. There are not so many in the meeting, so if others have questions, I think you can just ask them out.
Do you think other types of fiber prebiotics could have different effects? How do different alternatives work, or what do you know about the alternatives in this field?
Well, there's this is a sort of increasing field. So there are more and more alternative fibers and other components as well that is supposed to work prebiotic. And I think perhaps it would be might be a better strategy to use a mixture of fibers or prebiotic supplements, and perhaps also to use it together with some probiotics to increase the effect. Because you saw there was a slight increase in butyrate, but it was not significant, and obviously it was not enough to make the changes we were looking for in glycemic control and so on. So either the doses or the supplement we used were not good enough, or it's terribly complicated, you know, with this because we saw the variation, the inter-individual variation was much more important than the effect of the intervention, actually.
This is something, this is a very important and difficult topic. You know, we have experienced this several times, I think, with intervention studies that turn out to be a bit too small in the end because there's so enormous variation within the groups. And for you, of course, then microbiota wasn't the primary endpoint, but did you have any idea what kind of effect you were looking for when it came to GLP-1, for instance? Did you do any type of power analysis for this, or?
Yes, we did a kind of power analysis as that was our main outcome, but as nobody has done this indication that these prebiotic fibers may have a positive impact on glycemic regulation and GLP-1 response are mainly from animal studies. So we used a drug trial that showed impact on GLP-1 response, and we used that as proxy for the treatment that we were going to use, and ended up with that we needed a sample size of 24 to complete the trial. And of course, the reason why we went for a crossover design was just also to adjust for or try to avoid it at their own control. You know, the individual variation would not be that important when they are their own controls. So that was the reason why we ended up with that design.
There's a comment here from Ida, who has contributed. Comment to the negative effect on butyrate. Low abundance of butyrate producers at baseline might have influenced the final butyrate production. The changes in microbiota were also very small, and further analysis is likely to not give any statistically clear results with Picros or metagenomics. Yeah, perhaps.
That's good. You know, Ida had done this analysis. She knows much more about it on the potential than me.
Yeah. Is it possible to feed them butyrate, or will that taste too much of harsh butter?
Well, that's an idea. I had never thought of that, but that really is a good idea.
I don't think it will be a blockbuster, honestly. You have to have very good capsules.
Yeah.
Probably. Okay, any final questions from the audience? It's a final comment from Jonas André. Thank you for explaining, I guess.
On the question.
Yeah, I guess so. Okay.
Thank you for the talk, Anna-Marie.
There's a question here, yeah?
No question. I was just saying thank you for the talk. It was a nice and thorough presentation.